includes color. Okay, so more color effect. And again, this is true for this whole range of wavelengths that we're looking at. However, we just talked about the importance of this peak here at 260 nanometers, and 260 nanometers being the wavelength that is usually used to characterize nucleic acids. So what one can do is just put a sample of nucleic acids in a cuvette, set and put it in a spectrophotometer, set to 260 nanometers, and measure the absorbance. And let's say you have the absorbance measured at room temperature, 25 degrees, you get a certain value, whatever this might be, 0.3 or 0.35 or something like that. And then if you have a spectrophotometer that allows you to actually heat your cuvette, heat your sample, and you just follow the absorbance at 260 nanometers, you will see that from one measurement point to the next, you will have an increase in the absorbance at 260 nanometers. Okay? Up to a certain point where you do not get further increase of absorbance. Okay? What you have done is you have separated the two strands into single strands, utilizing heat, and you can say that you have melted the DNA. Okay? So this strand separation, in particular if it's based on heating of the sample, is known as melting. And if you then plotted your experimental results, okay, the absorbance values that you took at different temperatures, that you plot on your y-axis the absorbance as a function of the temperature, you would actually get a sigmoidal curve like that. Okay? So let's say you started at 25 degrees Celsius, you measured a relative absorbance of 1. Okay? If you get the same value, until you reach something like 55 degrees for this particular DNA molecule. And then you heat more, let's say at 60 degrees, you see already an increase, meaning that you have partial strand separation, and you heat more, you get data point here, and so on, until you reach actually a maximum value that you cannot further increase by heating the DNA more. And you have a plateau here. So pretty much you have a constant value here below about 55 degrees Celsius or so, and you have a constant value here, in this case above temperatures of about, 80-some degrees. Okay? So this is a classical sigmoidal curve. Can I have it? There's a classical sigmoidal curve that has a point of inflection here. Okay? Pretty much the halfway point between this constant level and that constant level, and we can use this point of inflection to determine a temperature on our x-axis at which we reach this point of inflection, this halfway point, and this, by definition, is called the melting temperature. Okay, and by definition, the melting temperature. Let me rephrase this. Melting temperature has been defined as the temperature where 50% of the strands are separated. Okay? So it's a halfway point between our native state and the denatured state. So by definition, okay, when we reach the melting temperature here, which is in this case, let's say 72 degrees or something like this, we assume that half of the molecules that were previously in a double-stranded state have been separated and are now in the single-stranded state. Now, if you think about it, but that's a definition, okay? If you think about it, it's questionable whether this is actually the case, that of number of n molecules that we have in solution, n-half actually has separated, and another n-half is still completely double-stranded. Or whether we have a double-stranded molecule and 50% of the sequence actually have been separated. So I'm not quite sure what is really the case. It doesn't really matter. By definition, we say, okay, that 50% of the molecules are now in the single-stranded state. Okay. I'm sure there have been some studies what's really going on. I think it's partial separation, but it doesn't really matter. Okay. So here we see actually two y-axes used. One is the absorbance. Okay, as I said before, we can plot the absorbance as a function of the temperature, and it's given as relative absorbance. Okay, starting with the absorbance we have at the beginning, whatever it may be set to 1, and then we see, okay, we have 30 some percent increase in absorbance in the denatured state. But it's also plotted here as percent hyperchromicity, meaning that in the native state we do not have hyperchromicity. Okay, so we go from 0 hyperchromicity to the fully denatured state where we would have 100% hyperchromicity, which we cannot increase further. And then we would have this point of reflection here at the 50% hyperchromicity value. Okay? So, now let's take a look at remelting curves that are rather different. The one in the middle actually has been obtained with a DNA that has, let's say, a statistical combination of AT and GC pairs. And we can measure a certain melting temperature, Tm here, and we compare this melting temperature with melting temperatures of a poly-AT DNA, meaning that the only bases that we find in this double-stranded molecules are adenine and thymine, okay, no G, no C, while on the right here we have a poly-GC, we only have cytosine and guanine, okay, no A, no T, and we see that the melting temperatures here, okay, this again would be the temperature value, which gives us the 50% hyperchromicity in these curves, is quite different. Why? That's a no-brainer. Yes. Okay, if we have poly-GC only, for each base pair, we would have 3 hydrogen bonds compared to only 2 hydrogen bonds if we only have A and T. Okay, so obviously our hydrogen bonding has an effect on the stability of DNA. So if you read in textbooks, okay, negligible, nah, not really. Okay, and here we can assume we have the same amount of A and T and G and C, so it's somewhere in between. Let's say statistically 2.5 hydrogen bonds. Now, often enough in molecular biology, so a subfield of biochemistry that we're dealing with right now, it's important to have an idea about the melting temperatures of double-stranded structures, double-stranded molecules. Typically, we're talking about DNA-based molecules. So, for example, if you want to amplify a gene, you could use a technique known as polymerase chain reaction. Some of you do this on a daily basis. Okay, so what you do is you actually use sequences that flank your gene and produce so-called primers. These are relatively short oligonucleotides that can hybridize with these regions flanking your gene. And then you play around with temperatures of your samples, okay, and utilize an increase in temperature to actually separate the double-stranded molecule to make your gene and one strand accessible for DNA polymerase, okay, and then you lower the temperature to a certain point where your primers, your synthetic oligonucleotides can actually hybridize with these flanking regions, and then you allow polymerase actually to synthesize your gene of interest in between. And then you just repeat this so often and you have quite an amplification of the sequence. So that's polymerase chain reaction in a nutshell. But it's sometimes quite critical what temperatures you actually use in this process, okay? And so you may have to have an idea at what temperature actually your primers will hybridize with the corresponding sequences in your DNA, and you can calculate this utilizing a number of methods, and I actually have one here. It's called the Wallace rule, which can be used for sequences between 14 and 20 base pairs, which just allows you to estimate melting temperatures. And this is based on the fact that there is a pretty much linear relationship between the content or the percentage of GC base pairs that you have in the DNA and the melting temperature. So since we have 3 hydrogen bonds between G and C, the more we have of these, the more stable more stable a structure we will form if we have more GC base pairs in a double-stranded molecule. Okay. So this relationship here is actually, interestingly enough, rather linear. Okay. What you need to realize is that so far we've only talked about one particular structure that DNA assumes, and I referred to it previously as the classical Watson-Crick double helix, which is also known as the bDNA. And this, for many years, I would say in this class, I really don't know why it's called the bDNA, not the aDNA, but then I did a little more research. Whenever I teach this stuff, I'm reminded of this, and then occasionally I have the time to look up things ultimately. And I think this is actually based on a identification system that Rosalind Franklin actually used. Okay. So the bDNA is a classical Watson-Crick helix, this right-handed molecule where it takes about 10 base pairs to complete one rotation. Okay. But this structure actually is only one of the conformations, one of many, many conformations that DNA can assume. And what you have to understand is that these pictures, these X-ray crystallographic pictures that were given to Watson and Crick by Maurice Wilkins and that had been obtained by Rosalind Franklin had been obtained from DNA crystals that actually had grown under certain experimental conditions. So I'm not quite sure how familiar you are with X-ray crystallography of biological macromolecules, but it's relatively tricky. Nowadays has become let's say quite a bit more advanced compared to the early days of X-ray crystallography. But you have to achieve to grow a crystal of a biological macromolecule, and what you do is that it's just more or less a trial and error exercise where you try to decrease the solubility of the biological macromolecule in a certain solvent. Okay? And you can decrease the solubility by adding salts, organic solvents, and things like this. And nowadays there are so-called kits that you can try, that you can go through, that give you pre-mixed solutions that contain all kinds of salts, for example. But in the early days, okay, they just had to come up with stuff and mix these solutions and then set up their samples in a certain environment. So the solvent that these biological macromolecules are dissolved in, or suspended in, is of great importance, but also the environmental conditions, and what's the temperature at which you crystallize, what's the humidity that these samples are kept in. Then another factor that is often not considered really, at least not rationally by people, is that it's a difference whether you have, for example, some shaking or something like this, or whether it's very, how would you say, what's the opposite of shaking? Stable environment, okay? Emotionally, I guess, stable environment. So some people think that there shouldn't be any shaking or any disturbance of the sample. Other people have found that, okay, if there is some vibration in the environment, that they may actually grow better samples. So there are a lot of factors actually, and it's very much based on luck whether or not you succeed in obtaining crystals. But the crystals that Watson and Crick actually utilized to come up with this aDNA had been grown at relatively high humidity, above 75% humidity. Okay, and there's nothing wrong with it, because in a biological environment, DNA would be in aqueous environment. Okay, so humidity is, what is it, 100%, pretty much? Okay. But it was found that if this humidity, this environmental humidity, is reduced, then a somewhat different structure is actually assumed by DNA. And this became known as the aDNA. Here we are. And we see that we obtain a molecule that is somewhat more wide, it's wider, okay? If we look at this virtual axis running through our molecule and we go back to our classical Watson-Crick helix, which is shown here on the left as bDNA, we have a certain tilt of our nitrogenous bases with regard to this axis that we go here from top down. But this tilt isn't really particularly dramatic. It's somewhere in the range of 6 degrees or something like this. We'll see some values here. What is it? 6 degrees, okay? But the tilting is much more prominent here in our aDNA. If we look at this sample, here is actually 20%. And I said that the molecule is actually wider. So instead of having 2 nanometers in diameter, 20 angstroms, remember, angstroms is nanometers times 10. We have 2.6 nanometers, so it's wider. Then also we need a little bit more bases actually to complete one turn. You see here that the base pairs per helical turn for the bDNA are given as 10.5. I said approximately 10. So 10 is usually the value that is utilized to do calculations. So that's what you should remember. That also is what Watson and Crick, I think, proposed. But later studies said it's more in the range of 10.5, but nobody really uses this. So it takes 11 for the A form actually to complete one helical term. But what these structures, the A and the bDNA, have in common is that they are still both